Hi, we present a computational framework for modeling emergence of color vision in the human brain. The paper was accepted to ICLR2025 as an oral presentation, and we are Atsunobu Kotani and Ren Ang from Department of Electrical Engineering and Computer Sciences from UC Berkeley. Please check out our project website, matisse.eeks.berkeley.edu for code, video, and dataset. So let's stare at this movie for a moment. This is our simulation of optic nerve signals, the only visual signal that our brain receives. Can you tell what you're seeing now? As you've just observed, optic nerve signals that come out of our eye is substantially different from our visual percept. So the question is, what are the neural mechanisms underlying these inferential computations? Traditionally, it's been thought that color vision is hardwired and neural circuitries define the exact color percept. In this work, we take a fundamentally different approach. That is, the cortex solves this complex decoding problem and discovers color vision solely from optic nerve signals. To prove our point, we will have three details covered: simulation of optic nerve signals, hypothesized cortical learning mechanism, and formal measurement of color dimensionality. Okay? Let me start with the simulation of optic nerve signals. We rely on the textbook model of retinal neural circuitry, the first steps in seeing by Robert Rodiak. For those of you who are familiar with the topic, we simulate midget private line pathway in 2-degree human central fovea. So how do we do that? We simulate cone cells, horizontal cells, bipolar cells, and retinal ganglion cells all together to simulate the output optic nerve signals. Let's take a specific example. So imagine you're now in the Louvre looking at the Mona Lisa. This image captures about 90 degrees field of view, and let's say this image of the Mona Lisa occupies about 10 degrees field of view. A human eye is never resting, and here we simulate fixational drifts and microsaccades. If we close up to the 2 degrees field of view, you'll see something like the movie on the right. We also compute cell positions using the data from Kirchhoff et al. 1990. Each white dot corresponds to a cone cell, color-sensitive photoreceptor cell, and you see the change in cell densities, dense in the center, sparse in the periphery. Each cell here receives this color spectral power distribution, and it is now the cone cell's job to process an incoming light. And this is what our retina, and more specifically cone cells, receive over time. Humans with trichromatic retina have 3 types of cone cells, as you see here. But this visualization is in false color. The actual signal that comes out of each cone cell is not color-coded, but rather a scalar value, as you see in this animation. The retinal process does not stop here, and to model the effect of lateral inhibition via horizontal cell connections, we apply a well-established difference of Gaussian's kernel. There should be on and off pathways, and it is decomposed into 2 separate signals. These signals are converted to spike trains, as you see in these movies, at the retinal ganglion cell layer. So here's our simulated optic nerve signals. When we compare this signal of a trichromatic observer with other variants, there's not much difference in these signals, as you see. This is noteworthy because it begs the question, how does a cortex discover different types of color vision purely from observing these optic nerve signals? Importantly, the cortex does not know a priori how many cone types are in the retina or whether the person is born colorblind or not. These 3 movies were actually simulated optic nerve signals for monochromatic, dichromatic, and trichromatic observer. And in fact, our simulation engine can take any image input, and here's the simulated optic nerve signals for various imageries. So we just talked about our simulation of optic nerve signals. Next, we discuss our hypothesized cortical learning. So our model says, look, everything is going to happen inside cortex, and it knows nothing else about the world except it receives the stream of optic nerve signals. Here we assume that cortex first time averages spike trains to get the time averaged optic nerve signals. Here we show only one image, but this model will learn from the database of hyperspectral images. And the key component of this model is that inside the cortex, we model learnable functions with millions of learnable parameters. So the question now is, what is the learning mechanism for the emergence of color vision? We say the cortex receives the optic nerve signals, goes through the learnable functions in order to make a prediction of the signal at the short time in the future. Now, of course, the brain receives the real optic nerve signal at the short time in the future, and it can compare its prediction against the incoming signal in order to calculate the cell-by-cell error in that prediction. The core of our hypothesis is that the brain makes use of this error to somehow update internal parameters used in making this prediction. In our simulation, we model this update as backpropagation with stochastic gradient descent as a functional placeholder for what might actually be happening biologically. Okay, so what's the learning mechanism? Our hypothesis is that the cortex learns to minimize the prediction error. But, how does the color vision emerge? To zoom in on this bold arrow here, making the predicted signal, can be factorized into the decoding and encoding process using these learnable functions. So the first step is to take the optic nerve signal and decode it into a person. It looks monochromatic now, but this is where colors will eventually emerge over learning, as I will show you in a second. Next, we model the cortex translates that percept at time t according to the analysis of the optical flow in the incoming optic nerve stream. Then, these learnable functions for decoding is invertible so that we can re-encode this translated color percept back into the space of optic nerve signals. And that's where we do comparison to the incoming optic nerve signal, getting a prediction error, and then we backpropagate to drive learning. What you're seeing here is the percept and the prediction error after 700 learning steps. This is after 1.2 thousand learning steps. You see that red-green color blindness has originally emerged with blues and yellows. And after 100,000 learning steps in this simulation, we get quite credible colors. Again, with no knowledge of the retina, except the signals coming down the optic nerves, the prediction error has fallen down to a very low value. Okay, so what was our hypothesis about how color vision emerged? The answer is that color vision emerges as cortex learns to minimize signal prediction error. Finally, the goal here is to make it as bioplausible as possible. So the question is, why is this percept in RGB color space? We made an important decision to model color in the brain as simply a high-dimensional vector, as you see here. By representing color simply in Rn, we say that, look, the brain actually needs to puzzle out the dimensionality of color in the incoming optic nerve signal. And indeed, we find that color emerges as a low-dimensional manifold in Rn space. But how do we visualize colors in Rn space? Here we use what we call neuroscope, which is simply a mapping from ND to 3D RGB color space with the minimum projection error. It's optimized as a visualization parameter independent of hypothesized cortical learning. Okay, so these are the results of different models after training. Remarkably, this mechanistic model actually predicts the right color dimensionality. For example, the model trained with this monochromatic retina results in 1D color vision, as you can see. If it's dichromatic in the retina, meaning there are two types of cone cells, the colorblind vision emerges, as you see. There are yellows and blues only. And if it's trichromat, the full 3D color vision emerges, as you see in this example. In addition, the squirrel monkey experiment with the gene therapy boosting is something we can model here as well. The simulation goes like this. First, we actually start training with a dichromat mosaic, this protanopia retina, as in adult male monkeys. And after that's converged, then we change some of the cone cells to the third cone type to simulate the effects of gene therapy. We then continue the learning mechanism with this new retinal configurations and the 3D color vision emerges, as you see here, at least in this model simulation. This result basically assumes that the hypothesized predictive learning mechanism is constantly happening in cortex as a tuning, even in adulthood, not simply as a development calibration process. Okay, the last column. This cortical model is agnostic to the dimensionality in such a way that tetrachromat vision emerges with full cone types in the array. Though we cannot visualize that the learn color percept with neuroscope, we can formally measure it with our simulation of color matching function test, which we call CMF sim. For those of you who are familiar with the topic, at a high level, what we are about to show you is just a classic Maxwellian color matching function test implemented in simulation. As I explained earlier, the cortex now decodes the optic nerve signal into a percept. And these optic nerve signals are generated from an input stimulus with our eye simulation engine. Of course, the input stimulus could be anything, like a gray color patch shown here, or this orange color patch on the bottom. What we do now is to fit them into the scheme of a classic color matching function test proposed by Maxwell and Grassman. The goal here is to find a match to the target light by adjusting the knobs of primary lights. Adjustments are made to reduce the difference between two percepts, and this is exactly how humans would do the color matching. Considering a trichromat observer, if we run this test with only two primaries, the error does not go down to 0, as you see in this example. But with 3 primaries, the error goes down to 0, as you see. These final coefficients actually define a slice of color matching function at 568 nanometer target light. We repeat the same test a number of times, and it generates color matching functions. Our results match with Stellis and Birch on real human subject. Great, so let's look at color matching functions with 1 and 2 primaries. If we overlay the color matching error, it clearly shows that 1 and 2 primaries cases fail to pass the test. The sum of these errors is exactly what each bar in this histogram shows, as 3 is the minimum number of primaries required to find a match to all target colors, which formally indicates that this model has 3D color vision. Great, so that covers everything in this result figure, and this is how we formally report that full cone types result in 4D color vision in this model. So far, we have quickly covered these 3 important details of our method. And now let us get into some technical details for our hypothesized cortical learning. This is a slide copied from earlier, and I want to start with this decoder function. Inside the cortex, we prepare what we call neural buckets for storing knowledge about the invariant retinal properties. It's not hard to imagine that having the perfect knowledge of all retinal functions that have generated the optic nerve signals, the cortex would have an easy time decoding that signal. For example, if the cortex knew about how lateral inhibition were applied, it can invert that process to generate photoreceptor activations or cone activations. Similarly, if the cortex knew the spectral identity of each cone cell, meaning each cone type, that will be useful to retrieve full color information. but these images are still warped. So one last thing might be positions of each cells. Then we can dewarp that image, invert the foveation to get the percept out. Of course, these retinal properties are not given and must be inferred solely from the optic nerve signal stream. Here's one possible initialization. Notice that lateral inhibition kernels do not have any of the center surround structure, and cone mosaic is all red, and cell positions are uniformly spaced. What was happening behind the curtain over the course of training is that invariant retinal properties are slowly but surely learned as the learning continues and finally converge to a solution. And we can reuse that knowledge, the inferred retinal properties, to convert the translated color percept back into the domain of optic nerve signals. Starting from the right, applying foveation using cell position knowledge, applying spectral sampling with cone mosaic, and then applying lateral inhibition with the inferred kernel. This is exactly how our model makes the next signal prediction. Okay, so that leaves off one last function we need to cover, that is this translate function. Now, let us reformulate the problem we are solving here. So when this image is presented at time t and t plus delta t, our eye model samples this stimulus from two different regions. We assume that this shift can be described as an eye motion dx dy, but our retina model makes the task quite complicated. Because of the foveation, you notice that spatial coordinate systems are different, xy versus uv. So the computational problem is that given these two images in uv coordinates, can we predict the eye motion dx dy in xy coordinates? Here, I blur the optic nerve signals to cut off high frequency details, and I think anyone can tell that this would be an easy task if you had the perfect cell position information and dewarped the image correctly from UV to XY coordinates. Why? Well, we can just do the search comparing these two patches and compute exactly how much one image is shifted from another. But the issue is that, as we saw earlier, the cortical model does not have the perfect cell position knowledge. Specifically, at the very beginning of the training, cell positions are initialized to be equally distant, resulting in no change in the coordinate system. Therefore, to predict the global translation or eye movement dx dy, the cortical system has to jointly optimize for the cell positions. Can the model succeed at this task? So, in ideal case, the model jointly learns cell positions and eye movement. Here I'm showing you how the correct solutions look like. On the left, I'm colorizing the cell density. It is high in the center where the fovea is located. And on the right, we get zero eye motion prediction error. Let's now look at our data. At initialization, predictions of cell positions and eye movements are quite inaccurate. But as the training proceeds, the cortical model succeeds at both tasks. The main takeaway here is that the model can infer the eye movement solely from studying optic nerve signals, and this result came out naturally from the same learning objective, predicting the next optic nerve signal. And this was the detailed cover for how our cortical model is implemented. Finally, let us end this supplement by showing you some of the results in motion. Here, we show the progression of learned visual percept. And as we saw earlier, it converges to 1, 2, 3, and 4D color vision for mono, di, tri, and tetrachromat retina model. We simulate variations of monochromatic retina, L only, M only, and S only. They all converge to 1D color vision. What about dichromatic cases? Protanopia and deuteranopia show blue-yellow hues, and it's teal and pink for tritanopia, and they all converge to 2D color vision. What about the gene therapy experiment? We first simulate dichromatic protanopia retina, let the model converge, and then we simulate the addition of L opsins. As soon as readaptation or relearning kicks in, the model converges to 3D color vision. And in this work, we also simulate to boost human trichromat to tetrachromacy by adding the fourth cone type. And we verify that the model converges to 4D color vision. And we have 2D to 3D and 3D to 4D color dimensionality boosting simulation results. In summary, this paper proposed a computational framework for modeling emergence of color vision in the human brain. We introduced the simulation engine of biological eyes, proposed self-supervised cortical learning purely from optic nerve signals, defined high-dimensional cortical color representations, and verified the color dimensionality using Neuroscope and CMF Sim. We view this paper as an invitation to the research community to explore and improve our proposed framework. And here's the team and support and our acknowledgement. Thank you very much for watching.